Okay, and all right, I'm gonna turn off my video to save some resources. And okay, let's continue. Okay, so you can see my screen, right? All right, perfect. Okay, so last time, we talked about we learned a lot of new terms, very important terms in quantitative genetics. So hopefully uh, you have had chance to review this stuff uh, over the weekend or when you have time, because we have so many terms, definitions. They are very confusing. So hopefully you can clearly uh, uh, differentiate those terms. So last time we learned how to define the genotypic value, right? So that's the expected phenotypic value, uh, uh, the difference between the expected phenotypic value with the midpoint. So this is how we define the genotypic value for three different genotypes in a F2 population. And so given the Hardy-Weinberg equilibrium theorem, so we can calculate based on the allele frequency, we can calculate the expected value for the population mean, expected value, genotypic value, expected genotypic value for the population, which is defined as the mu g. And this is how we use the formula to calculate the mean for the population. And uh, not only we learn how to calculate the expected value, we can also calculate the variance of a random variable, right? So we use the formula to calculate the variance of the genotypic value, which can be expressed using this formula, right? And we also learned the average effect of an allele. So here we talk about a biallelic population. We only deal with two different alleles, A1 and A2 from F2 population. F2 for a F2 population. And so this is how we define the effect of a allele. For example, A1, and so this is the final expression for A1, right? So similarly, we can define alpha 2, which is the average effect for the other allele using this formula. Okay. Okay, now, so if we define alpha 1 and alpha 2, so we can take the difference between these two and define this as a alpha. So alpha is the difference, actually, it's the difference between the two allelic effects. And we call this term as the average effect of the allelic substitution. And these two formulas are actually showing how you can relate each individual allelic effect with the allelic substitution alpha here. All right. So those are only like involving algebra, so not a big deal. And okay, now we we need to learn some new concepts. So very important. So here we would like to learn the breeding value. So what is the breeding value? So here, so breeding value is the value that we calculated by summing the two allelic effects for a specific genotype. So for example, so the breeding value for 1, here, the big A represented the breeding value. So don't confuse this term with this, okay? A1, A1. And don't confuse this term with A1. So we use A1 to represent the first allele, right? And we use A1 to represent the genotype for this genotype, right? But here is A11, so it's different. So the difference is very subtle, but this is the place where we usually get confused. So don't confuse this term with the other two, right? Okay, now A11 represents the breeding value for genotype of A1. And it is defined as the, because in this genotype, when we want to calculate the breeding value for this genotype, so this genotype involves two A1 alleles, right? Each A1 allele will have a average allelic effect from that allele. So that's why we take the sum. So we add up these two A1 specific allelic effects. So that gives you the breeding value for this genotype, which is A11. Is that clear? Okay. All right. So because we have derived the expression for A1, so you can easily rewrite this formula into this using the allelic substitution here. Let me clean the slides a little bit. All right. Okay, so the same. So here we can calculate the breeding values for three genotypes. So here A12 means the breeding value for the genotype of A1 and A2. And A22 means the breeding value for the genotype of A2 and A2. Is that clear? So this is how we define the breeding value. This is a very important concept in academia and in industry, because in industry, in some company, for some company that which are specialized for developing the breeding skills, and for example, Monsanto or Pioneer, so they use this term a lot because they try to optimize the cultivars with the desirable breeding values. So, all right. Okay. Now, so here, so it's very clear to in this table. So in this table we can clearly see, right? Okay. So the difference. So this is the difference between the genotype of the genotypic value for the genotype of A1 with the population mean, right? And this, yeah, this can be expressed by the two allelic effect from A1 plus an error. So there's an error term here. All right, the same thing here. So for this genotype, we can express the difference between the genotypic value for this genotype with the population mean by using the sum of the two allelic effects from the second allele plus a error term here. The same thing for this two, right? These two are the same. So here we have one allelic effect from the first allele and the other allelic effect from the second allele. Because think about like, so these two alleles come together to form this genotype, this progeny. So one allele carries the effect of this, and the other one is carrying the effect from here, right? And they come together to form this, but still there's a deviation between this individual with this genotypic value. So we define this as the error term here. Is that clear? Okay, very good. So, or you can look at here. So we define this error term for the first genotype, which is A1 by this. So if you treat this as the difference between the genotypic value for the first genotype and the population mean, and this include two parts. Actually, this include two parts. One part is the breeding value for this genotype, and the other part is the error. So if you take this difference, this will gives you the residual, the deviation for this genotypic value, and the same for the second genotype and the third genotype. We can also define this deviation using the difference here. So it gives you different expression of the deviation or error. And if you look at these three terms, error terms, okay, these three terms only involved what? The D, right? Not A. So this deviation from the breeding value, so the deviation of the breeding value from the expected genotypic value is due to the dominance effect. So here we give this a name called the dominance deviation here, right? And okay, so this is a little bit complicated. I don't expect that you can fully understand all this stuff here in the class right now, but so take some time to review this, to read the book, and so because so since this is the benefit for taking the online course, because we record every course, so, and I provide the link, so if necessary, so you can go back to the link to review the lecture to make sure you understand this, right? So this table summarize what we have learned, the concepts here. So we have three genotypes for the F2 population. And this is their frequency given the Hardy-Weinberg equilibrium, right? And this is how we define the genotypic value for these three genotypes, right? And this is the breeding value we just defined here. So this is how we define the breeding value. And this is the dominance deviation. We can treat this part as the error from the model. Error term from the model. We're going to talk about the model shortly. All right, so right now, so we want to define these terms here. Any questions so far?
One question.
Yes, please.
Well, I got a couple. So the value of 2 for the breeding value, for the error terms, the dominance deviation. Why is that value 2 there?
You mean here?
Yes, because it makes sense for the breeding value, why the value of 2 would be there.
Okay, that's a good question. I think we need to do a little bit of math to find why there's a 2 there. So let me see. So okay. So let me show you. So oops. Okay. So if we want to see why this is this, right? So we need to find what is this and what is this. So here, it's easy to find the A11 is here, right? And it seems like, okay, there's a 2, but how about here? Let's see if we can find another 2 to make it more, to make this, to make sense to you. Let's go back. So this is let me see. Oh, sorry. Okay, 511 minus UG. So what is that? 511 is this. Minus UG is this, right?
Mm-hmm.
So if you calculate the difference between these two, so what do you get? So there's a single A here, right? There's an A here. So we're gonna take A out. This is common thing. 1 minus P1, but plus P2, right? Is that correct? If you put them together? Okay, now this becomes minus, right? Still P1, P2, and D, right? Okay, what is this? This is P2 again, right? 1 minus P1 is P2. So P2 plus P2, this term will become 2P2 A minus 2 P1 P2 D, right? Is that correct? So this is the first term.
Yeah.
Okay, now remember this. I cannot remember this, but help me. So 2P2 times A minus 2P1 P2 D. So let's go back here. So 2P2 A minus what? 2P1 P2 D, right? That's this part.
Yeah.
Okay, now we minus this. 2P2, but this is alpha, not the A. So you have to use the alpha, so which is very complicated, I think. Let's see, alpha is this. You use alpha to replace here. I believe you're gonna end up with this.
Okay, I get it. I'm getting it. All right. It follows the same structure as all the other terms that we've been introduced.
That's right. Yeah. So this is how we teach the class. So I try to build up this. I try to set up the small bricks first, and then we just put the little small bricks together to build a building. So what I have introduced today was totally built upon the stuff we have learned last time.
Mhm.
All right. Does that
Right.
make sense?
Yep, they all follow the same general structure of algebraic math.
That's right.
So-
Yes.
Okay, I got it.
All right. Okay. Let's see. This guy. All right. Okay. Let's continue. So, very good. Okay. Now, before we want to move forward, so I'm gonna introduce. So I believe most of you have already learned this from some statistical courses, but here because I still want to spend a little bit of time to refresh your memory about the linear model. So what is a linear model? So here, so first, the linear model is a simple model that we can use to evaluate the potential association between two random variables, right? So usually, if we talk about the linear model, we usually can use this model. For example, y is one random variable. We use b0, which is the intercept, plus b1, which is the slope, times another random variable, right? And you can put an error term here. So the error doesn't matter because, yeah, all right. So this is a linear model. So linear model is used to evaluate the potential association between two random variable of interest. So here is an example. So we have two random variables here. So the first one is the temperature of the day. The other is the y here is the sales of the ice cream. So suppose you are a ice cream salesman in Disneyland. Definitely, yeah, right now, because Disneyland in Anaheim is closed, so it's not a good timing for you. But yeah, in the past, so if you are a careful salesman, you want to find if the sales amount, the money you earned every day from selling the ice cream in the Disneyland to lots of happy children. So you want to find if there's association between the day temperature and the sales, right? So if you are very careful salesman, so you just log your data every day, and this is the data you get. So suppose you worked for probably like 15 days. There are 15 data points, right? For each data point, you have a paired value, right? X and Y here. So X means the temperature, day temperature for that day, right? And Y is how much money you earn from selling uh, the ice cream, all right? Okay, so this is the data. So what do you think about your data? Do you believe there's a potential association between these two variables? Do you believe? Yes, of course. So, but just by looking at the data, you cannot make a conclusion. That's not the way our statistician do, right? So we have to prove. Using your data, we have to prove there's a association behind these two random variables, right? So what we can do is what? So we can fit the data with a model. We can fit the dress, fit. We can fit the data, and we can fit the data with a model. We can fit the fit yourself in the dress to see if the dress fits you well, right? So this is what we have learned before. All right. So what we can do is we can propose a model, linear model, and try to describe the data using the model and to see if this model is significant or not. So this is what we're going to use for future test to test the association between what. So can anyone tell me? So we learned this linear model association here. So we want to use this linear model to test what in the future? Think about the class. So we're talking about the genotypes and we're talking about the phenotypes. So what do you want to test using linear model?
We want to test the null hypothesis.
Yeah, that's right. So yeah, your answer your answer is correct. But I want to the point that I want to ask is like we want to use the linear model to test the potential association between any single locus and the phenotype of interest, right? Suppose we want to see if there's a specific locus, we want to see if the variability or change in the genotype at this locus is associated with the variability of the phenotypes, let's say the rice yield. So we can do the same thing, right? We can use the linear model to test if there's association between the genotypic variation and the phenotypic variation. So that's the test for a single locus. So if we can make the test, so if we can scan this kind of test, we can test the genome repeatedly by scanning the genome, by scanning each locus on the genome. So that's the GWAS, right? So GWAS is actually we repeatedly do the test of the association between a single locus and the phenotypic variation. So if we scan the genome by testing each locus with the phenotype, so then we can possibly identify the genomic loci that are responsible for the trait, right? So this is the GWAS. So if you do not do not have that background about the GWAS, so some people saying something about the GWAS, so yeah, you probably think, yeah, that's a very, very advanced technology. But if you understand the underlying theorem, so it's pretty simple. So we just use the regression model, linear model to test the association between each locus and the phenotype. And then we extend this analysis by scanning the whole genome using the same strategy. And we can use this to identify genomic loci that are responsible for a traits. So that is GWAS. Is that clear? Any questions?
Could you go over that one more time?
I'm sorry?
One more time.
Yeah, one more time. Okay. So here, so in this example, we just tested the temperature X, right? If the temperature X explain the variability in the daily sales, right? So if there's a trend, it means, okay, the temperature indeed determine how much money you earn by selling the ice cream, right? Okay, now, for example, if this is a specific locus in the genome, we want to see if the genotypic variation, let's say A1 here, A1, A2 here, or A2, A2 here. We want to see the variability or change in the genotype at this locus is associated with a phenotype, let's say the yield. Yield, let's say Y1, Y2, and Y3, right? Okay, suppose this is not the locus that is contributing to this phenotype. You're gonna see what? You're gonna see the plot is a flat. You don't see a regression line here. If you put the genotype here, A1, 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 A2, and A2, A2, right? This is the Y, the yield. So if there's no such association, you're gonna see the point are pretty flat. Is that right? However, but you cannot stop here, right? There are so many markers on the genome, so you can repeatedly test each locus to see if they are associated with the variability of the phenotype, right? So probably for most of the tasks, you're gonna get the same plot here, like everything is flat. But if you test the locus, which is indeed contributing to this phenotype, what do you get? You're gonna get something like this, right? Do you? Right? And this will gives you what? Gives you a conclusion. Okay, bingo, we found the locus, or we found the SNPs, or we found the SNPs within the gene, which may be responsible for the phenotype, right? Okay, so we just repeatedly use this linear regression model to test along the genome until we find, so this is a GWAS. For example, this is the chromosome 1, chromosome 2, chromosome 1, chromosome 2, chromosome 3, chromosome 4. For human, we have like 23, right? And X and Y, something like this. So you can generate a plot like this. So if there's no signal, no association, you're gonna get statistic this low. But once you get a signal, so a loci that is responsible for this phenotype, you're gonna get a peak here. Another peak, not that high. Another peak, right? So if you scan the genome and do this repeatedly, do this test, you can easily, so if you set a threshold, so you say, okay, if the peak go beyond this threshold, then this will be a significant test. So this test can help you to identify the loci that are associated with the traits. Is it clear? Very good. So yeah, this is GWAS. I believe most of you will be very happy to learn this, because in the future you're going to use this powerful tools to help you advance your research. Right? Okay, any questions? Okay, now back to our ice cream. ice cream problem. All right. Okay, so all right, so probably in high school you have learned. So if you want to definitively define a regression line, so there are two parameters you need, right? Two parameters. So one is the intercept, I just mentioned here, and the other one is the slope. The slope is here, right? Slope. Right. Okay, and can anyone tell me, so if you want to test whether there's association, significant association between X and Y, the two random variables, so what do you need to test actually? What do you need to test? The slope, right? So we need to test if there's a slope. If the slope equals to 0 means, okay, the X is not a determinant of Y, so there's not a there is not a association between X and Y, right? However, if the slope is not equal to 0, or in the other way, let's put another way. So if the slope is significantly different from 0, then this is equivalent to the statement that, okay, X is associated with Y. Does that make sense? So the slope is the most important thing in a linear model. So let's focus on this. All right, so how do we calculate the slope, or how can we estimate the slope and the intercept? All right. So we have learned in the past. Where is my? Okay, it's here. Let me remove this. Okay, so suppose we have data like this, and we want to summarize the data using a linear regression, which is here, right? So we have intercept and slope here. So how do we estimate these two parameters here? So there's a famous method called the least square estimation. So what is the least square? So this is what I would like to introduce. So actually, to summarize this data using using a regression line, so there are multiple ways to do this, right? So in this plot, it gives you the best regression line, but you can use different way to summarize. So you can use this line, or this line, or this line, or this line to summarize your data, the relationship between X and Y, right? But which one is the best, all right? We want to find the best line, regression line, that can summarize the relationship between X and Y. So this is the criteria. The name tells you the criteria, the least square estimation. So we need to find the best, the optimal regression line that go through all the data with the minimum least square. So with the minimum difference between the data points and the regression line. So what does that mean? That means like, okay, so there's a line go through your data, and when you check the difference between each data point and the regression line. So for example, this is the difference between the first data point and the regression line, right? So this is the second difference, the third difference, the fourth difference, and so on, the fifth difference. So we just check the difference between each data point and the regression line. So we want to see which line, regression line, gives us the smallest squared difference. Does that make sense to you? Does that make sense to you? So this is how we estimate the regression line. We try to minimize the difference, okay, between each data points and the regression line. So, and this is why we name this method as the least square estimation. Okay, very good. Now, go back to our problem. So here we have our data. So our data is what? Is the genotypic value. So we need to find the best way to summarize this data. So that's the regression line. All right, the regression line actually represented the breeding value. Now we need to go back to our breeding values. We have 3 genotypes and we have 3 breeding values, right? So this point is the breeding value for the A2, which means the A22. And this, the circle line here, circle dot here is the breeding value of A11. And this circle dot is the A12. Okay. That, oh, sorry, A12. Okay. So these 3 breeding values are on the regression line, and the difference between the real data, the observed genotypic value, and this breeding value is called the dominance deviation. So we have spent a little bit of time on these terms, right? The dominance deviation. So that's the difference between these two, right? So you can find, so this is a very useful, actually, this is a very useful diagram. So I hope you can spend time to understand this, because this diagram shows you the relationship between the different terms we have learned so far. So for example, this is the population mean here. And this is the average effect of the allelic substitution. So by understand this, you can probably find the link between the different terms we have learned so far in the quantitative genetics. All right. Okay. Now, if some genes do not show the dominance, so if for some genes there's no dominance effect and there's no dominance deviation, then we call those kind of genes, that kind of genes as the additive genes, because only like additive effects are involved. There's no dominance effect, right? So just 1 minute break, our brain break. So far, do you have any questions?
So the we have the the actual data points, we're treating them like breeding values.
Breeding values actually are the expected thing. So the breeding values are on the regression line.
Okay, that's helpful.
Yeah, the open dot.
Okay, so the regression line is our breeding value.
Yeah.
The actual values is the actual genotypic value that we get.
That's true, yes.
And then the difference from the breeding value and the genotypic value is, it says the average effect of allelic substitution, but you used a different term.
Oh, you mean this one? No, this one, this is alpha. Okay, so see the difference between the third breeding value and the second breeding value is the alpha, and the same here. This is alpha 2.
Okay. So that's the differences between breeding values of specific alleles, or
that's true. Yes.
Okay. And then the difference between the actual data point, or the genotypic value, and the breeding value on the regression line, that is our error. That is our error, or dominance deviation.
That's right. So yeah. So exactly. So we're gonna talk about this because when you try to predict the breeding value for the next generation. So suppose you have a breeding population, right? So we want to use this current generation to generate a progeny, and you want to use the genotype for the current generation to predict to produce the breeding values for the next generation. So the breeding value is the thing you can predict. You cannot predict the real phenotypic value for the next generation because of the error, because of the dominance deviation, because the dominant deviation cannot be predicted using the regression model. Only the additive, only the breeding value part can be predicted.
So you can't actually predict the dominance deviation using this.
That's right.
That,
yes.
that is something you need the actual physical data for.
Yes, exactly. So yes, and another thing is like because the breeding value is, so if you look back to the breeding value definition, you're gonna see breeding value is only involve the additive part, but the dominant part actually represent the interaction between two different alleles. Usually when we define the dominance effect, we are talking about the heterozygous, right? So if you see like the two alleles, A1 and A2, so that might be a dominance. But if you look at the two homozygous individuals, so we do not assume there's a dominance effect, right? Yeah, so that's why. And that might be the limitation for the linear regression model, because we can only predict based on the additive effects, because the dominant is something like troublesome. So that's why we put them in the residual or the dominance deviation. Yeah. All right. Okay. Any other questions? Okay, now, so let's study the genetic variation components. All right. So first, when we look at the phenotypic value, so G represents the expected phenotypic value, right? So I hope you still remember. So, all right. And suppose we have different genotypes, and we can calculate the variability or variance within. We can calculate the variance of the expected phenotypic value among different genotypes. Okay, so we want to see among this total variation, so we call this total variation, total phenotypic variation, phenotypic variation. We call the variation in here as the total phenotypic variation. We want to see, so what is the component? So can we decompose this total phenotypic variance into different components? So this is how we want to study this. All right, so first we want to see, because we know the total phenotypic variation should be coming from two places, right? One is the additive effect, and the other one is the dominant effect. So here, A represented the breeding value, and D represented the dominance deviation. Okay, so now let's look at the expectation of this breeding value here. So this is what I required each of you to know very well. So if I ask you to calculate the expectation of something, so you can immediately come up with the equation, right? So expectation of X. So in the test, in the final exam, I'm gonna ask you to do the calculation for me. I will give you a random variable and the associated and the possible outcomes values for this random variable, and the probability that is associated with each value of this random variable. So if given that, you need to know how to calculate the expectation for this X for me. So that's the formula, right? So I summation of all I xi, all the value that xi can take, multiplied by the probability which is associated with that value. Is that right? So this is how we calculate the expectation. All right, so if I ask you to calculate the expected value for the breeding value for me, so that's the formula you need to use, right? So we have how many breeding values? We have 3, right? We have A11, the breeding value for the first genotype, A12, and A22, and this is the probability for that genotype, right? So, and this is how we calculate that. Is that clear? Any questions, any problems here?
Can you remind me of the nomenclature? So A is obviously breeding value, and then the P value, and then the P's we have here, that is the value of probability.
Yeah, probability. So let's go back to this slide. So
okay, I got it.
Okay, here. So the A11, a P11 means this, right? The probability in the Hardy-Weinberg population. The P11 means the frequency or what is the percentage of the individual in this population carrying this genotype, right? And because this is the probability, and the A11, not A1 means the genotype, but the A11 is the breeding value for this genotype, right? So then we come back to here. Oops. So this is the breeding value for this genotype, and this is the percentage, the proportion of this genotype in the population, right? So this is exactly meet with the formula I just showed you, right? To do the calculation expectation. So we have 3 genotypes. That's why we calculate the sum of these 3 terms. So this is how we calculate the expected value for the breeding value in the population, right? F2 population. Any questions? So, okay, so when you do the calculation, it looks scary, but once you simplify this, that's easy, 0. The expected value should be 0 because this is the way we define this. The expected breeding value should be 0, right? And also, you can use the formula to calculate the variance of the breeding value, right? The variance of the breeding value. Why is this? Can anyone tell me? So when we calculate the variance of X, let's say variance of X, this equals to expectation of X squared, right? Minus the expectation, minus the square of the expectation, right? This is the formula. But in this case, we have already calculated the expectation of X equals to 0. So this term is gone. This term is gone because the expectation of X is 0 here. We just proved this, right? So that's why the variance of A breeding value is just equal to the expectation of the A squared. All right. Okay, so this is how we calculate the expectation of the breeding value in a population and the variance of the breeding value in the same population. And we can do the same thing to calculate the expectation for the dominance deviation, right? Which is also 0. And we can also calculate the variance of the dominance deviation in the population, which is this. And so in the past, we have demonstrated, so we have used the same similar formula to calculate the variance of the expected phenotypic value, which is equal to this. Now, let's see what happened. So this, let me see. Okay. All right. Oops. Too messy. So if you add up these two things together, this will be exactly equal to this. Yeah, you can do this by yourself. Yeah, you can just, because here we have alpha, so you have to use the expression of the alpha here, and then you plus this two thing, and this will gives you the identical results here. So which proves that the variance of the total variance in the phenotypic variation can be decomposed into two portions. One variance component due to the breeding value, which is the additive effects, and another component, the variance of the dominance deviation, which is due to the dominant effects. Okay. Okay, let's move on. All right, so now, based on what we have learned so far, so here, so this is how we express the observed phenotype, right? The observed phenotype Y equals to the expected phenotype, sorry, the expected phenotype plus the random error. And this expected phenotype can be written into two parts, additive parts and the dominant parts. So if we calculate the variance of the total phenotypic variation, so we can write this total phenotypic variation into three parts. So this is the variance due to the breeding value, the additive. This is the variance due to the dominance effects, and this is due to the random error. So this is how we calculate, we decompose the total phenotypic variation into three parts. All right, now here we go. We have two important concepts in genetics, not just in quantitative genetics, in genetics, especially good for the breeding. So we have the broad sense heritability here. So the heritability, so first, heritability. Heritability means of the total, so when you study a trait, so if you study a trait, so you can easily calculate the total variation of the traits in the population, right? So when you study a population, for example, F2 population, and if you are interested in a trait, let's say the rice yield, all right? And you can easily calculate the simple variation of the yield, right? The traits. So that's the total phenotypic variation. All right, but you want to see as a geneticist or as a breeder, so you want to see what's the proportion of this total phenotypic variation in rice yield can be exploited by genetics, which means what's the proportion of the variability you observed in the phenotype can be are inheritable, can be exploited by genetics. Is that clear? So this is how we define heritability. So for example, if you see 100% of the variability in the rice yield, but only 20% of this, usually for yield. So I have analyzed a lot of like rice project, and the yield, total yield, is not a good predictable traits. The predictability for that trait is usually around like 20% or 30%, which means the heritability of the rice yield is about 30%, which means only 30% of the variability in rice yield can be exploited by genetics, is inheritable. Is that clear? Okay, so mathematically, how do we define the heritability? So the broad sense heritability is, so so this is what the denominator is, the total variation, right? Total variation in the phenotypic value, and the numerator is the variation that can be explained by genetics, either the additive effect or the dominant effect. So if you take this ratio, so only this part cannot be explained by genetics, right? The environmental variation. So this proportion will give you the broad sense heritability. But as I just mentioned, because when you do the prediction for the next generation, we can usually do good job on the breeding value, the additive part, but we cannot have a good control on predicting the variability due to the dominance effect. So that's why in breeding program or in genetics, quantitative genetics, we often use this narrow sense heritability, which is the among the total phenotypic variation, what is the proportion of the total variability that can be explained by the breeding value, the additive effects. Am I clear? Any questions?
So why is narrow sense eliminating the dominance effect?
Okay, good. Oops. Let me make, so let me erase the messy handwriting here. Okay, good. So this, yeah, this is a good question. So I may give you a question like this in the final. So tell me about the difference between these two terms, the broad sense heritability and narrow sense heritability. Okay, so this is why. So yeah, this is how I would like to answer your question. Okay. So ideally, we hope we can 100% predict the variability due to the genetics, right? So this is our hope. So we hope we can build a perfect model that can be used to predict 100% of the variability due to genetics. Because for the environmental part, we can never do any prediction. That's the random error. So that's why. So this is the ideal situation. So if we can develop an ideal model to predict both additive effects and the dominance effects, then that's perfect, right? But in reality, I just mentioned, because there's a limitation for the linear regression model, we can very well handle the additive effect, but we cannot at the same level handle the dominant effect. So that's why we put the dominant effect deviation into the error term. So so this is ideal situation, ideal. But unfortunately, this is the reality. Okay, so in reality, the current model, linear model, can only do a good job in predicting the additive part. So that's why this narrow sense heritability is the most commonly applied in industry and in research. So this is why. And the broad sense means broader, which include the dominance effect, but the narrow means we eliminate the dominance effect from the numerator. Does that make sense?
Mm-hmm.
Mm-hmm. Good question. So yeah, pay attention to this one. So the probability for me to give a problem like this in the final exam is very high. Okay. What time is it now? Okay, we have another 20 minutes. So good, we're doing good job. Any other questions? Okay, so let's do some math calculation here. All right, so far in this chapter 5, we have we started from the Hardy-Weinberg equilibrium, right? And So we learned the general situation, like if we have two alleles, A1 and A2, if they do not have the equal frequency in the population, so what happened? So we based on the general guideline situation, we derived a lot of concepts. So that's why in those definition you see different P1, P2, etc. Right? But in F2 family, so this is a very special case. This is not a general case. This is a special case. So why? Because in this special design F2 family, so P1 equals to P2 equals to 1/2, right? Because we start from a single F1. So think about just one locus. So the F1 heterozygous, and we start from that single genotype to produce the F2 family progeny. And in this F2 progeny population, the two alleles, the frequencies for these two alleles are equal, right? 1/2. So this is a very special case. Okay, so if P1 equals to P2 equals to 0.5, so then it's easy. So we can easily calculate the genotype frequency as this, right? Does that make sense too? All right. Now, so if this is the case, and then just use the formula I showed you before, right? So you can easily calculate. Oops, what is here? Sometimes I lost the trace of my cursor because the point is too small. All right. So now, if you use the formula to express the alpha, which is the allelic substitution, right? And then you replace these numbers with the genotypic frequency, then you're gonna simplify this. So in F2 population, in F2 family, the allelic substitution is identical to additive effect we defined at the beginning. All right, these two things are different, right? Don't confuse them with each other, all right? So alpha, only in F2 population, alpha equals to A, which is the additive effect. And so in this case, if you calculate the variance of the phenotypic, the total phenotypic variation, so equals to this. Any problems here? So I think, yeah, this is easier, right? Because just now we talk about the general case. Because in general case, so you cannot have these nice numbers. You have to use the P1 and P2. So that made the equation messy, right? But now, since we have a beautifully those beautiful numbers due to the simple situation, they have two progeny, so we can simplify this equation like this. So, any questions? Okay, good. Okay, now, so little by little we try to migrate. So I hope, like, so this is my style. So I try to, like, sometimes when I introduce new stuff to you, so I will use some examples, but finally we will come back to our own problem, and we try to find the connection between the previous knowledge and the current knowledge, right? So here we're doing what? We're doing linear model. We try to use a linear model to do what? We try to use a linear model to test the association between a locus and the phenotype. So we want to see if the variability in the genotype at a specific locus is associated with the phenotypic variation. All right, so this is what we want to do. So we focus on this line. Although we talk about a lot of stuff, definition, diverse of the definitions, but we will always come back to this point. We're gonna generate a linear model to help us test the association between any given locus and the phenotype, right? All right, so this is how we do this. Suppose we're talking about a special case, F2 family in a Hardy-Weinberg equilibrium, right? And so this is the linear model we can use, right? This is the linear model. Okay, because we're looking at so we want to test the association between the phenotypes here. So this is the expected phenotype, and this is the intercept. So so think about the regression line. So this is equal, the mu here is actually the B0, which is the intercept. And this is the first X. So additive effect. So the additive effect is B1 here. So this is the first slope that we want to test. And this is the X1, which is used to define the random variable. So this is the random variable. Z is the random variable that we use to define the genotype for the additive effect. And this W is another random variable that we use to define the genotype that is associated with the dominant effect. So this is B2. So in this X2 here, so in this regression line, actually we have two slopes. So I don't know if you can get the point. So usually we have a single line that way. For that line, we have an intercept and a single slope. But here we have two slopes. So just expand your imagination. So instead of using a two-dimensional, we use a three-dimensional, something like that. So in this regression model, we have two slopes to test. One is for the additive effect, and the other one is for the dominant effect. Both can represent the association between the genetics and the phenotype, right? Both, either one can represent the association between this locus and the phenotype of interest, right? Okay, now let's see how we define the first random variable. So remember, the Z is a random variable that we would like to use to define the genotype for the additive effects. Okay, look at here. So because we have three genotypes, right? So if an individual takes the genotype like this, so there is a additive effect, right? So we put a +1 here. And this is a F2 population. So the associated probability for this genotype is 1/4. So any problem here? Any problem for understanding this here? No? Oh, okay, very good. Okay, now, so when the genotype takes this, so A1, A2, so is there any additive effect now? No, that's why we put it 0 here, right? And for this one, there's a minus additive effect. Is that clear? So this is how we define the first random variable. And the second random variable is the same way. So if we look at this genotype, so A1, this is a homozygous, so there's no dominant effect, right? So we put 0 here. The same for this homozygous genotype as well. For either of this, the probability is 1/4. Only one the genotypes takes this form, A1, A2, there's a dominance effect with the probability of 1/2. Is that clear? So this is how we define this two random variable for three different genotypes, right? And we can put this two random variable into the same model like this. Okay, so I think it's easy if you have some background in setting up a regression model, but I think yeah, probably it will be a little bit not straightforward for you if you do not have the experience in dealing with the multi-regression model. But yeah, so because I don't think I have time to go into the details, but I hope when you have some time after class, review the slides, read the book again, and maybe you can do some exploration online to find how we define the random variables for a regression analysis. And this can help you better understand the content here. Okay. All right. Because yeah, so another thing is like, so this class, although it's very much relevant to statistics because it's a statistical genomics, but this course is not taught in statistics department, but in genetics and the plant sciences department. So because the student, graduate student, just like you, come from different backgrounds, so that's why sometimes like I also refresh the very basic knowledge in statistics to help everyone to be on the same page. So I think probably, yeah. So as I mentioned, if you have had some experience in dealing with regression analysis, maybe, yeah, this part is not that hard. But if you do not have the experience in dealing with the regression analysis, so yeah, please review this part and read the textbook and find some useful information from online to help you better understand how we set up a linear regression model. So because here, this is very important, because in the future, when we talk about the GWAS, genomic selection model, we always use this simple linear regression model to help us to do the hypothesis testing. So this is very fundamental, but very important. So make sure you understand what we are doing now. Because the very important example is today. So I show you the ice cream and the sales, right? And the temperature. So basically we want to test the association between two random variables. This is exactly the same in the quantitative genetics. So think about the locus is just one variable, X, right? Each locus can take different genotypes. That's one variable. And another variable is the phenotypic value. So that's the thing we are interested in. So we try to test if any locus is associated with that phenotype. So this is the way that we can scan the genome to help us to identify the genomic loci that are responsible for the traits of interest. So we will repeatedly apply the linear regression model in genome-wide quantitative analysis. All right, any questions? Okay, all right, so on this slide, oops, where is it? Okay. This is how we define these two random variables. And this is, we just showed this, right? So in the F2 population, so the variance of the phenotypic variant, the total variance of the genotypes is equal to this and this in the F2 design. And we can also use the formula to calculate the variance of Z, so the first variable, which is used to code the genotype for the additive effects, and the W is the variable that is used to code the genotype for the dominant effect. So this is how we can calculate. So in the F2 population, the variance of the Z equals to 1/2. And the variance of W equals to 1/4. So you can figure it out by yourself. Just use the regular formula for calculating the variance of any random variable, right? And because here Z and W is not correlated, it's not associated, so that means like the covariance between them is 0. So that's why we can simply add these two things together. Why is that? So the next slide shows you, okay, so this is how we calculate the variance for a random variable, right? So I just showed you the variance of the random variable x equals to the expectation of the x squared minus the square of the expectation of x. This is the formula for calculating the covariance between two random variables, x and y. So formula like this. So when you use the information about the F2 population, you can easily prove the covariance between Z and W is equal to 0. So just use this formula here. So, okay, so this slide shows you why in F2 population, if you design your Z and the W in that way, so how we can prove the covariance between Z and W, sorry. Let's see. This slide shows you how we can prove the covariance between, this is the covariance between Z and W. We use this to denote covariance between Z and W equals to 0. So if you follow these formulas, you can prove this. Yeah, it's not that hard. This is only for the F2, right? Because in F2, we have a special setting. What is that? The P1 equals to P2 equals to 1 over 2. Okay. All right. Oh, we are very good in terms of in terms of timing. So, oops. Let me see. Okay. So far, any questions? Okay. All right. Any questions for today?
Will this, we won't have to be tested on this particular proof that you just showed us.
Mm-hmm. Sure.
We won't.
Okay. Let's do this. Okay. This one, right?
Yes.
Mm-hmm. Okay. So here, yeah, please do not forget about here we are dealing with a special case, not a general case, right? Special case means, okay, F2. We are talking about F2, and in the F2, so when we look at a single locus, the two alleles, the frequency are equal, right? P1 equals to P2 equals to 1 over 2. So by doing this, you can easily use the formula to calculate the variance of the Z variable. So we just defined that Z has 3 values, right? Z equals to 1 for the first genotype A11, right? A1. And the second value for Z is 0 when the genotype is A1, A2, right? And for the third genotype, which is A2, the value for Z is defined as -1, right? And the associated probability is easy, right? So 1 over 4 for the first genotype, 1 over 4 for the third genotype, but 1 over 2 for the heterozygous genotype, right? So this is how we calculate the Z, the expectation for the square of Z. So that's why we put a square here, right? This is this term. And now we talk about this term. This is the square of the expectation of Z. So what is this? This is exactly what we calculate the expectation for a random variable, right? So Z can take 1, 0, and -1 with probability of 1 over 4, 1 over 2, and 1 over 4. So if you add up these 3 things, that's the expectation of Z. And we put a square Finally, that's the square of the expectation of the z. And this part is the expectation of z squares. So make sure you're not confused with these two terms. Okay, this is it's not easy. So I remember when I first started learning statistics, I was so lost, so confused. So don't worry. So if you feel lost, don't worry, because everybody the same. So if you practice more, if you read more, and once you get familiar with this, you will feel comfortable about these concepts. All related to your experience or exercise practice. So here, this is the definition. So if we want to calculate the variance of this is the variance of a random variable z. So this is the formula, right? So the variance of z equals to the difference between two things. The first is the expectation of z squared. The first term is the expectation of the square of the random variable. So which is the z squared. So we want to calculate the expected value for z squared. That's why we put z squared here. So this is the three possible values for z squared. And we want to calculate the expectation of z squared. Then we're gonna do the summation of these three possible values for the z squared multiplied by its associated probability. Is that right? So this is how we calculate this. But here is, so this expression actually should be, so it's easier to write in this way. So this is easy, right? Expectation of z squared. If you calculate the expectation of z squared first and then calculate the square, that's this thing. That's why you see different. So within the parentheses, you see the general expression of for calculating the expected value for z, right? Because z can take three values here: 1, 0, and -1 with these probabilities. You just sum up these three values multiplied by its associated probability. That's the expectation of the z square. And finally, you just calculate the square of the thing. That's the second term. And the whole thing, the whole calculation, just complete the calculation. Oops. Complete the calculation for the variance of the z. So this is one thing. And the other thing is this, right? So you can also do the same thing to calculate the variance for w. And now the last thing you need to prove if the covariance between z and w equals to 0. So this is how we calculate the covariance, right? So look at here, this is easy. So we can calculate the easy. Oops. Oh, there's a so there's a typo. Sorry. Probably you are referring this. So there's a typo here. So this should be expectation value of the z multiplied by w minus the expectation of z multiplied by the expectation of w. Yeah. Okay. So this is easy, right? So this is how we calculate the expectation of z, expectation of w. So we have already calculated this part and this part. And now we just worry about this part, right? So this part is here. So we want to calculate the expected value for z multiplied by w. So z multiplied by w, there are also three possible values. So when for the first genotype, z equals to 1 and w equals to 0, that gives you the first value for z times w. If the individual takes the genotype of A1 and A2, so in that case, z equals to 0, but W equals to 1. So this gives you another value, right? And if the genotype is A2, so Z equals to -1, but W equals to 0. So this gives you the third possible value, but all of them are 0. And this total, the entire thing here is 0. So, and the second term is here. So because EZ equals to 0, so this gives you another 0. So 0 plus -0 equals 0. So you complete your proof. The covariance between Z and W equals to 0. So is that clear? Mm-hmm.
Yes.
Okay.
I didn't want to interrupt you, but I was asking whether we would be like tested on this specific proof.
Oh, you mean test? Oh, okay. So, so I probably will not, because our test is a open book, so everything is here. So maybe I will not. Let me think. So is there any way that I can test you to prove this? Maybe yes. So maybe I can give you a different way to define Z and W, and then I will I will ask you to calculate the covariance of Z and W to see if that kind of design gives you the 0 covariance between Z and W. No, you don't like it? Okay, thanks for the feedback. So I'm gonna rethink about that. Yeah, but yeah, so in the past so it is challenging for us because I don't know, because this is my first time to teach online. So I don't, even we have like open book test, but I don't know how I can organize this test through the Zoom. So I need to talk to other experienced faculty to see how can handle. But I think although it is challenging, but not particularly challenging for us because we are open book test. So so I think it should be okay. But for this specific problem, so I don't think I just give you a question to prove this because this is too easy. You just copy this and you write down the answer, right? But yeah, I'm gonna think about some other alternative ways to test it if you understand how we can check if the covariance between two random variables is equal to 0 or not, right? But basically, if you understand how we calculate these things here, so don't worry, you will be okay. You'll be good. You'll be good to answer that question, right? So any other questions?
I have one.
Yeah, go ahead.
Can you post us some practice questions for the expectation calculation?
Say that again. Can I post what?
Some
I'm
practice
sorry.
questions for the expectation calculation.
Oh, so yes, sure.
Okay, thanks.
No problem. So I think so right now it's still early, right? So we do not have midterm. We only have a final exam. So probably like 2 or 3 weeks before our final exam, I'm gonna post some practice test for you. So in that way, so in that way you can learn what you would expect to see from the final exam. Does that sound good?
Yes, thank you.
Sure. Okay, no problem. So yeah, we will do that. Right. Don't worry. So take it easy. So this class, I know this class we learn some difficult, very solid stuff. But in terms of testing, I'm not gonna put too difficult questions on the final exam. So don't worry about this. As long as you understand the logic, usually in the final, when you see the questions on the final, you're gonna see those are very simple numbers, and I just want to test if you understand the logic behind. So even for the simple numbers, so when you do the problem, just show me each step in the between. Even eventually you did some incorrect calculation, giving a wrong numbers, but it's okay. If I can see if I can clearly see your logic for deriving the final answer, you can still get the full score.
Okay.
Don't worry. So
Thank
in
you.
terms of the knowledge, this is a very tough class, but in terms of testing, this is not that hard. This class is okay. All right. I hope so far you still enjoy the class. Yeah, hopefully every one of you can learn some useful things for your future study, because yeah, this is a very applied class, and you're going to learn a lot of useful things, especially for some fundamental but very basic important concepts in statistics and quantitative genetics. All right, so if you do not have questions for today, let's stop here. And I still hear strong wind outside. Scared me. All right, so good luck everyone. So hopefully I can still teach my class sitting here on Thursday. Yeah, I don't want to be evacuated from my area. All right, take care. Thank you, bye-bye.